Hello students, this is Ashta K. Sanuj, Assistant Professor, Department of Microbiology, EMA College of Arts and Science. Today we'll be talking about the genetic basis for antibody diversity. So this will be a big lecture, so I am planning to make a couple of series for this topic. So let's get started. Antibody diversity. Well, you know that antibodies are antigen binding proteins, right? Antigen binding proteins which are present on the B cell membrane. B cell membrane, and they are secreted by whom? Plasma cells. Now, all the antibodies, they share structural features, structural features are same for all the antibodies, though they are diverse according to the antigen that caused the generation of that particular antibody. Now let's see what actually is antibody diversity. Now, you know that there are millions of antigens, and our immune system has the ability to produce specific antibodies against all the antigens. Now, this diversification in the antibody production, that is the diversification in antibody production is known as antibody diversity, right? There are more than 40,000 genes in our genome, and these genes code for all kind of proteins, say for enzymes, for regulatory proteins, and you can say IG, immunoglobulin, right? Now there are only few genes in our genome that code for immunoglobulin. Now let's see how it is done. Okay, that is what we are going to learn. Now you understand what is antibody diversity. As I have told you, like your body is capable of producing antibodies to huge variety of antibodies. So this series I will try to explain you like how what makes that antibody so diverse that it can produce any type of antibody for any kind of antigen. Okay. Okay. So actually to support these things, certain theories were actually proposed by different people. Let's see a few of them. The first one is the germ line model or theory. This theory accounts for the high degree of antibody variability found in the population. According to this theory, which suggests that every B lymphocyte has all the gene, all the genes, sorry, for every type of immunoglobulin, but transcribes only one. So this is what the germline model or the germline theory states, that every B cell will have whole genes for the I mean it has all type of genes for every type of Ig immunoglobulins. Okay, it is full of genes and it can be any genes, but it transcribes only one. Later on, another theory was put forward, which is known as, again, somatic variation theory. Now, according to which few inherited genes undergo extensive somatic alterations to generate the thousands of genes that give rise to myriad of antibodies. Okay, now this somatic variation theory states few inherited genes undergo extensive somatic alteration, or you can say somatic hypermutations, to generate the thousands of genes that give rise to. So it is going to give thousands of genes, and this thousands of genes is going to give rise to the myriad of antibodies. Now, basically, now we know these two theories are not accepted, right? Now, there is another theory which is called as two gene model hypothesis. Now, it was put forward by Dreyer and Bennett in the year 1963. Now, what they suggested was there are two separate gene which codes for which encode single immunoglobulin chain. Okay, immunoglobulin chain. So these two separate genes which was mentioned was V region, known as the variable region, and the C, that is the constant region. So it has got two separate genes for these two. And when these two are brought together, Okay, they are brought together under the DNA level to DNA level to form continuous message, or you can say that is a process of recombination. Now, what happened? It actually it is transcribed as single immunoglobulin gene. Okay, so this is what is the two gene model hypothesis, which was proposed by Dreyer Bennett in the year 1963. He said there are two separate genes which encode for the single immunoglobulin chain. The two separate genes were one for V region and the other one is for the C region. When these genes should come together at a DNA level to form a continuous message. Okay, so that's a process which I mentioned is recombination, and it gets transcribed as single immunoglobulin genes. They propose that more than 100 to 1000 V region genes in germline cells. And there are actually only single copies of C region, single copies of C region and this single. So these are the few theories which actually try to explain the antibody diversity, why it is so diverse. So here, if you see, the first thing is the germline model or germline theory, where actually they suggested that every B cell has all the genes present in them, but it can transcribe only one. Okay, that is how the diversity has appeared. Now, next theory, which states that is a stochastic variation theory, few inherited genes they undergo extensive mutation and then they create thousands of genes which will help the diversity of antibody. So we very well know that these two are not accepted at this um, in today's life. Now, the another theory which was put forward was two gene model hypothesis, which was proposed by the Dreyer and Bennett, and he's quite reasonable. He said that there are two separate genes which encode for a single immunoglobulin. Now, these two separate genes was one for the V region and another one is for the C region. But only thing is they have to be brought under in such a way they have to be transcribed. They first have to be brought together in a DNA level to form a continuous message, which has to be transcribed into a single gene. Then they also stated there are hundreds of thousand V region genes in the germline cells, but only a single copy of C regions was found. Now let's move to something more interesting. Another theory. So it was in the year 1976. Tony Gawa and Hazoum actually. They both said there is a direct evidence for separate genes encode the V and C regions. Okay, he said there is a direct evidence for separate genes encoded which encode the V and the C region of immunoglobulin. Now, these gene segments are rearranged in the coat of B cell proliferation and enabling only a small number of genes to produce a much larger number of antibody. Okay, so he also said that the gene segment are rearranged. You can say reshuffling also, rearranged in the case of, in the course of, sorry, it's course of B cell proliferation, enabling a small number of genes to produce a much larger number of antibodies. Okay, now he has done an experiment which I would like to explain it here. He has taken myeloma cells and also embryonic cells. What are myeloma cells? They are cancer of plasma cells, okay? Now here, both of the cases, the DNA was isolated and the DNA was isolated in both the cases, and then they were subjected to restriction endonucleases, restriction endonucleases for the digestion. So now they have formed small fragments. These are the restriction fragments, right? Restriction fragments. Now he took these and did electrophoresis, and he found out, if you just take, okay, this is for myeloid cell and this is for embryonic cell. I just had a single DNA and two DNAs here. So what did he conclude from this experiment? So let me just explain again that he took DNA from the myeloid cells and embryonic cells. Then they were digested with the help of restriction endonucleases. Now the restriction fragments were subjected to electrophoresis. And then after the experiment, he concluded that chromosomal DNA is no longer identical to the germline DNA. Yeah, it was not identical with the initial germline DNA. Then that there had been reshuffling, as I mentioned, reshuffling or rearrange during the B cell differentiation which allows create a wide range of antibodies. So these are the theories which actually try to explain the diversities of antibody. Okay, now let's go to the next session about the gene organization.